Hello everyone, today I will talk about plant conservation. Simply put, it is about protecting plant and their DNA so they can survive diseases and climate change in the future. But here's the problem. Traditional methods like just building fences or protecting habitat, we cannot see the invisible health of a plant. Even a forest that looks green might actually be weak inside because of inbreeding. We need to know their genetic health to truly save them. So why do we need genomic and molecular tools? First, high precision. These tools work like DNA fingerprints to find hidden details that our eyes cannot see. Second is faster result. This genomic technique gives us result in just a few days, which is much faster than waiting years for plant to grow. Third is species resilience that help us to identify the strongest plant so we can make a better decisions for their long-term survival. That's all for my part. Now my teammate will explain about the applications. Thank you.
Genomic in the plant conservation, type of genomic, nuclear genomic, application, population genomic and world genomic excavation to assess genetic diversity and inbreeding. How does it help plant conservation? WSG allows research to locate specific homozygous region, run of homozygosity that included inbreeding by identifying harmful mutation. Conservation can design genetic research models such as control pollination between populations with different detection load to restore fitness and prevent local extinction. Importance of the plant composition complete blueprint. Unlike other types like chloroplast DNA, the nuclear genome provides a complete historical record of a species, including past population, the present and recovery. An example of the nuclear genome being used in the plant conservation is the genomic discourse and management. A critical entry. Genomic in the plant conservation, type of genomic, chloroplast genome, application, phylogenetic exploitation and species identification, DNA barcoding. How does it help plant conservation? The chloroplast genome evolves more slowly than the nuclear genome, making it ideal for resolving complex relationships between species or ligands. This application helps define conservation uniquely important in plant conservation. The chloroplast genome is highly stable and evolves slowly, functioning as a reliable DNA barcode. This allows conservation to easily identify endangered plants and discard them from similar-looking common species. Tracking ancestry and genetic health can be inherited through only unparameterized inherited. Example: species identification, forensic table analysis, legal evidence, and biodiversity monitoring.
For the first molecular marker application, I will discuss the use of simple sequence repeat or microsatellite in genetic diversity assessment. SSR markers are highly polymorphic and condominant markers are used to assess genetic diversity and population structures, especially in endangered plant species. The variation in the number of repeat units allow research to detect heterozygosity and alert variation within and between plant population. This helps identify populations with low genetic diversity and inbreeding risk, supporting conservation breeding and restore program. For example, SSR markers are very used to study genetic diversity in rainforest tree species, help to guide conservation and management decisions. The second application is DNA barcoding for species identification. DNA barcoding uses standardized gene regions to accurately identify plant species. The method is especially useful for morphological similar species, earlier growth step, and process plant material. By comparing DNA sequence and DNA distance, DNA barcoding enables species-level discrimination. DNA barcoding helps prevent misidentification and illegal repayment for endangered species. It also supports law enforcement biodiversity monitoring, and overall species protection. An example is the identification of endangered medic plants use a BCLA mark and its2 marker.
The significance of genomic and molecular marker approach in plant conservation lies in their contribution to more effective and informed conservation effort. This approach provides accurate information on genetic diversity and population structure, allowing conservationists to identify vulnerable populations and reduce genetic erosion. As a result, this support better decision making in both in situ and ex situ conservation strategies, ensuring long-term sustainability.
So overall, the use of genomic technique and molecular marker analysis has greatly improved how we approach plant conservation. With these tools, we are able to identify plant species more accurately, especially when morphological features are unclear. They also help us understand genetic diversity and population structure, which is important to direct inbreeding and genetic enrollment at an early stage. Because of this, conservation decisions are no longer based only on physical traits, but on genetic evidence. This allows conservation to design more effective in situ and ex situ conservation strategy, including breeding and restoration programs to ensure the long-term survival of endangered plant species.